Hi, my name is Imroze, and I'm an assistant professor of biology at Ashoka University, which is a newly formulated university which came at Sonipat near Delhi. So in today's talk, I'm just going to take a slight diversion from the different topics, wonderful topics about that we have been hearing since the morning. I would like to take you to a more fundamental of evolutionary biology of how immune responses might respond to pathogens and how the immune response can be optimized. Since we have been facing pathogens, it's one of the most continuous selection pressure that we all have been evolving with. And Tribolium is one of my model system that I typically use in my lab. So as the title suggests, it says the infection history drives the evolution of divergent immune responses and mutation rates. These are seemingly two different part of my story. I'll try to touch upon both of them if the time permits. But I think without wasting a little more time on it, let me just quickly get into the real topic of my work. So first thing that I'll use to define infection history is something that we all grapple with, and yet we don't pay that much attention, which is the co-infection. Right. It is very difficult to find in nature that one host is often infected by one pathogen. This typically we always see multiple pathogens are infecting a given host, and host has to balance its immune response to figure out how to respond different parts of immune response. When different kind of pathogens might elicit different forms of immunity, and our host responsibility is to invest or at least show immune responses which are carefully balanced so that it can cater the need from both the pathogens. Well, there are many such examples. HIV, for instance, it's famously it's known to co-occurring with hepatitis C virus, influenza with severe infection of many of the influenza patients they are known to be infected on pneumococcus. Of much recently, even the SARS-CoV-2, it's now been widely known to co-infected with several other bacterial infection also, which kind of exacerbate the health condition of the given patient, right? To give you more example, this is a kind of statistical data which has been collected over many years. It has been found typically this chikungunya, dengue and Zika viruses often cohabit in a given environment. And as a result of that, many patients who have been diagnosed with one eventually were found to contain multiple viruses. So you see that different viruses might ask for different immune responses, and how the patient has to respond efficiently to stay healthy or at least to survive, even though they're grappling with the same different infections. So the key question that I would like to know, and which is again, co-infection as a topic of research has been addressed by some people, mostly at the molecular scale, but yet we atrociously know very little about how a host might evolve an immune response, given that, as I said, the populations are grappling with multiple pathogens at one point of time. Can we evolve an efficient immune responses against multiple infection? If so, how it is different from a given single infection that we typically know of? Most of our textbook examples, most of the papers, they often look at one pathogen, one host perspective, and we know a lot of information about that. What we are trying to know, at least what we are trying to know from my work, is to find interactions between different pathogens and interaction between immune responses that could be elicited by different pathogens. Okay, so do hosts evolve divergent responses against infection caused by a single versus co-infecting pathogen? So to do this, I have been using Tribolium castaneum, not largely driven by its value as a storage grain pest, but rather its suitability as a interesting model system. Because if you follow the habitat of Tribolium castaneum beetles, they typically live in silos in a moist environment, and which is a breeding ground for multiple microbes, multiple pathogens. So this naturally, these beetles are known to be exposed to different pathogens at one point of time. So my guess was, before we started this work, we thought this could be an interesting model system because they are naturally known to responding to multiple infections very frequently, right? So and of course, as I was trying to outline, that beetle survival might thus rely on their ability to mount, this is a very important one, a balanced and effective immune response. It's not about very strong immunity. After COVID, we are almost all of us aware of that, having a strong immunity is not a solution. Many of the patients have been showing symptoms because we typically showed excessive immune response to a pathogen. As a result of that, many people had shown post-disease symptoms, right? Many of the disease symptoms were manifested after months, okay? So important is to know how to evolve a balanced and effective immune responses. Okay. So just like as I used a suitable model system, I also had to choose two suitable model pathogens. So Bacillus thuringiensis, as you all know, this is a very famous bacteria and have been typically known to be a superstar insect pathogen, right? And we also had a Pseudomonas entomophila. This is again a natural insect pathogen. So we thought of looking at beetles' perspective when they deal with these two infections. And there are interesting facts about Bt and Pseudomonas entomophila. Bt is a gram-positive bacteria, which necessarily involves a different kind of immune response. Let's say insects have multiple pathways like toll receptors, toll pathway, or IMD pathways, different pathways. What is important is to know that Bt, since it's a gram-positive, it might induce toll pathway activation, whereas Pseudomonas entomophila is a gram-negative pathogen. So it has its own known set of immune receptors, immune effectors to induce with. And when we did a very first preliminary preliminary experiment with the beetle infected by Bacillus thuringiensis or Pseudomonas entomophila or a mixed infection of both, we typically found this interesting phenomena. At a given dose, at certain doses, it looks like, right? So if you see the red line, this is a mortality trajectory when they're infected by Pseudomonas entomophila. And this is how they look when they're infected with Bt. Now, by looking at this graph, it must be telling you that their infection dynamics is very different, right? It looks like Bt, to deal with an infection caused by Bacillus thuringiensis, the host has to induce certain fast-acting response because the killing trajectory is very quick. It's within maybe 12 hours after infection. Whereas Entomophila, the red line, it takes time. So host has at least some time to recover and carefully think, well, I want to induce this kind of immune responses to deal with P, but they cannot afford the same against Bt. Okay, and if you look at the bacterial growth dynamics of these two pathogens, if you look at the Pseudomonas entomophila, which I'm calling PE for the rest of my talk, if you look at the bacterial load, within 24 hours, there's a sharp rise in bacterial load, but then it falls down and it remains in the host. It's like creating a persistent infection in the host. However, if you look at Bacillus thuringiensis, it looks like a rapid clearance. It kind of reflects how it responds to its survival also. The mortality is very quick and hosts are also clearing the infection very fast. Right, so in a way, I can safely say that, well, their growth dynamics, their infection dynamics are likely to be different. Right? So now the question is, if you infect the beetle with these two different pathogens, what would you expect? Now let me give you a quick overview about insect immunity. Again, insect immunity has a lot of similarity with the higher organisms. Their innate immunity is like to be conserved across taxa. So therefore, at least insects can serve as a very interesting model system to understand innate immunity and their evolution. Right. So let's say you think about a fast-acting pathogen like how Bt was, right? It is creating rapid mortality, rapid clearance, and to deal with such pathogen, host has to activate certain immune responses which are also fast-reacting. And we know there are some like that. Let's say, for example, phagocytosis. This is again the so the insect, they have a cellular immunity. They have something called blood cell-like molecules which try to eat the bacteria. Phagocytosis, this is a Greek word derived from a Greek word called phagine, something that eats. So this eating thing is a fast response, and it also has a response called melanization. This is again a conserved immune response that we also have in humans, right? We produce melanin, and an important thing about these two pathways are fast, but they also come with a super physiological cost. When a host induces phagocytosis or melanization, they also produce a lot of cytotoxic molecules, a lot of harmful molecules. They're accompanied with production of a lot of reactive oxygen species, Now, I don't have to reiterate that production of lot of ROS can also be harmful to your body, because ROS can create direct damage to our cells and organs and even our DNA, right? However, if you think about a pathogen like Pseudomonas entomophila, which is a slow-growing pathogen, and typically we know that there are a different group of molecules called antimicrobial peptides, which are typically synthesized in the host to deal with slow-acting pathogens. So we had a fair bit of background information before we started this experiment. And now the remaining part was to look at their evolution, right? What is to understand the relative importance of phagocytosis, melanization activity versus antimicrobial peptide secretion. This is where the mechanistic details are going to converge into. Okay, so to understand these evolutionary properties of immune system, we typically adopt a strategy called experimental evolution. So those who are not aware of this method, this is basically, we literally recreate natural selection in the lab. Experimental evolution is a method which gives you or allows you to study evolution as it happens in the lab. There has been many landmark experiments. Most famously, there are a lot of microbial evolution. You can literally see a bacteria evolving antibiotic resistance in a test tube, perhaps, right? We are replicating the same principle using these beetles. What we do, let's say let's say some young 12-day-old adults, they were infected with a pathogen, and now after infection, some of them will die, and we then take the live individuals and resume the generation afterwards. So it's a continuously selecting for individuals who are better able to survive after infection. And this process goes on and on and on, right? This is how typically natural selection would work, right? Okay. And we had different populations. For example, sorry about the formatting error. It was shifted from a Mac, and it's showing a little bit of ditches. So there are one set of molecules, one set of beetles who are naive individuals, the individuals who were not exposed to any pathogens. This is a control, one kind of control. We also had another control called sham infection. Here, the infection was caused by a buffer, right? This is a procedural control. And then subsequently, we had 3 experimental populations. One in which beetles were across generations, they're infected with Bacillus thuringiensis. Second was where they're infected continuously with Pseudomonas entomophila, and the third is the co-infection one, mixed infection one, right? And this experiment has been continued for almost 21 generations now. We are continuing the experiment for last 2.5 years, right? So now these beetles are living with these pathogens for 21 generations, and we already have started seeing some very interesting patterns already. Okay, so this is what it looks like. Look at this trajectory. These lines are suggesting how the beetle mortality after infection changed across generations. So the red line is the populations which have been infected with Pseudomonas entomophila. Green lines are the population which have been infected with the mixed infection dose, right? And the blue line are the beetles who have been infected with Bacillus thuringiensis, right? So this is how the data look like. This is a response after 21 generations, and looks like the response has been dramatic, right? So at least at one pathogen, beetles could easily evolve super resistance. You could see they're already percentage survival, y-axis, if you follow this one, the beetles infected with Pseudomonas entomophila, they're almost now becoming super immune, super resistant to the infection. Whereas, the Bacillus thuringiensis, the blue one, are still hanging around, somewhere around something around, where they started with, right? Whereas if you look at the green lines, it is also showing a very delayed response. So it is only after generation 16 the mixed infection doses responded. The beetles infected with the mixed pathogens, they evolved some sort of resistance. So now this data gives us 2 important insights. One, that the beetles have one collection of immune system, but they're responding very differently from different pathogens, right? So response against 2 pathogens can be fairly divergent. And the mixed infection is somewhere it looked like as if the response to mixed infection has been carefully calibrated by both the pathogens. So now it is coming somewhere in the intermediate, right? And this is an interesting part, because since we have been dealing with multiple infection quite a lot in the natural world, it looks like the efficiency of our immune system to infect or being infected and to evolve against a mixed infection can be fairly different from what would they have been evolving against a single pathogens, right? So this is a different time points where this is a time around generation 7 when Entomophila started diversifying from the other populations, and this is an interesting point where the mixed infection responded around 17 to 18 generation. We have data till 21 is not showing here, and we could see that green line is still going upwards. So it has already started picking up its resistance, right? So evolving a response in co-infecting pathogens could be less efficient compared to something where they have been infected with a single pathogen. Right?
How many adults do you take in each generation?
Oh, it's almost around 400 adults. So it's a fairly large population size, right? So this is where I assayed these beetle populations individually. You could see that Bacillus thuringiensis selected lines do not show any change in the resistance. You see the blue lines is a control regime where they have not seen the infection, whereas the gray line is the one where they have seen the infection. So, and none of them show any difference. We also measured their ability to clear infection. So y-axis gives you bacterial load or pathogen burden, and you see that none of the control or either control or BT populations, they don't show any difference. Whereas when we looked at, again, reflecting what we have seen in those graph when multiple generations together, this is an assay which was done around after 18 generation of selection, right? And you see that red lines are the Entomophila selected lines, and they have shown a very strong response, right? And simultaneously, we could see that Entomophila population has also evolved a stronger bacterial clearance ability. We also see a response against mixed infection dose. It shows a response after at generation 18, and again the better. So this graph kind of suggests that the ability to survive better against infection actually coming from their ability to clear the infection. So it has the bacterial resistance what has evolved in these populations, right? And we have been doing this experiment fairly thoroughly. We have been tracking the evolution of these responses across generation 2, generation 8, generation 13, generation 18, and we plotted the response for across different generations. We found a very interesting depiction of the graph. Let's say, it just shows that how divergent the rate of evolution could be. The red line is the Entomophila selected lines, and the green was on mixed infection lines. Ultimately, they're possibly going to the same fate, but response has been taking to different pathways altogether, right? So, now the question is, why did the population infected with the Bacillus thuringiensis, they did not evolve resistance? We have been finding very thoroughly that whenever beetles or insects, they have been infected with Bacillus thuringiensis, as I said, they are likely to induce fast-acting toxic immune responses, and that's exactly the case. We find that compared to the control, the infected beetles, they have a higher phenoloxis activity. Now, phenoloxis activity could be pathogen killing, but at the same time, it also induces a lot of immunopathological consequences. As a result of that, even though it is beneficial for the host to evolve stronger, this phenoloxis activity, so one could assume that had they been evolved higher amount of phenoloxis activity after evolution, they could have also cleared the infection more efficiently, but it didn't happen. And possibly because this super cost, the cost of producing very high amount of phenoloxis response is constraining them. This is a result where it shows that beetles where they have evolved, beetles where they have higher phenoloxis activity, they actually show a very reduced secretion rate from the kidney. So this is a malphizian tubule activity is their kidney activity. And it seems after infection and after inducing phenoloxis response, their kidney function goes to take a pitfall, right? So it looks like, any stronger expression of phenoloxis activity could make them completely non-viable. So this is possibly the reason why BT selected lines did not evolve. Whereas when we did some of the RNA sequencing, RNA transcriptomic analysis with our beetle population, we have been, and this is a very basic data that we just got the data a few days back. These experiments are still in not a complete mode. It's just a evolving story I just thought of sharing with you. And from whatever pilot experiment that we have been doing with these beetles, we found there is already finding some of the interesting molecules that are being overexpressed in the population which have been infected with Pseudomonas entomophila. So I'll just briefly describe what these molecules are. It don't have to pay attention a lot of this thing to complex names. But if you look at this particular model which we are slowly formulating, it looks like the Entomophila infected populations are showing high evolution of higher expression of inducible immune activity, and this is what we actually predicted also at the beginning, because this is a slow-acting pathogen. So organisms have the luxury to induce a slow-acting inducible response. Sure, right? Response. So it kind of matches up with our prediction that the organism would evolve some sort of inducible kind of immune responses. It also shows that they also have increased a ROS-mediated pathogen clearance. This is a slightly interesting observation, because at the same time that they have been evolving slow-acting inducible molecules, they also have ROS-mediated. But a basal level of ROS production is perhaps important for the organism to sustain. Because they are also important part of our immune system. And interestingly, we also are finding evolving mechanism to control their immune responses. This is an important, IMD pathway is an important pathway of immune expressions when hosts are infected with a gram-negative pathogen. And it seems that they're also evolving a better regulation. So, any of the time, having our uncontrolled, excessive, suboptimal immune expression is not at all desirable. Right, so they're also evolving a tighter regulation on the immune responses. Well, these are slowly forming stories, and if I just have to summarize of what I have been telling so far, we found a divergent rate of adaptation to single versus co-infect pathogens. We are now tracking the similar molecular signatures in our mixed infection populations also, to see how these populations are coming in between their single pathogen cousins, single pathogen population controls. Right? It looks like Entomophila has evolved strongest response, followed by mixed infection, and BT still hasn't evolved a response, and we are waiting for them to evolve, right? And of course, the mixed infection populations, they have been showing a critically balanced immune responses, which again, and which is again somewhere falling in between what they have been evolved for this respective individual pathogen. Is coming somewhere in between the Entomophila and BT infections. Right? We are currently working on the mechanism supporting the fast evolving versus slow evolving immunity in these populations, and at the same time we're also trying to figure out getting more mechanistic support that whether indeed involvement of more cytotoxic immune arms against BT is restraining or constraining its evolution or not. Right? So how much time I have? 8 minutes. Okay, so I'll just, so this is one part of the story, and I'll just quickly tell you another part of the story which we have been following up of recently, and it's just at the surface, we're just scratching the surface. So this is an interesting story which has been started by one of my PhD student, Bashubi, and a postdoc, Dr. Joy Bose, and here we are trying to link immunity with deleterious mutations rate. Inflammatory responses are notoriously known to produce, as I said, a lot of reactive oxygen species, a lot of cytotoxic intermediates, and besides damaging the cells and organs of the body, they also create damages to the DNA. Many of the cancers are known to have, they're known to show a lot of aberrations in their DNA structure after inflammatory responses have been gone through. So, and we found, well, now, forming the mutations, the generation of a mutation has not been a very novel thing because it has been there in the literature for long. But what we found, these mutations, if they're happening in the germ lines, of the organism, after infection, after inflammatory response has gone by, those mutations can transform into their offspring. And if this process goes on, it can create a mutational load on a population. So this is what we have been finding so far. I'll just quickly take you through since I don't have much time. So this is what I was trying to tell you. If you have a stimulus, you will be having an immune activation, and many of the inflammatory responses, mostly known in many of the metabolic diseases, are known to create mutations, right? For example, if you think about inflammatory molecules, since they produce, reactive oxygen species, reactive nitrogen species, they can create DNA damage or genomic aberration or DNA lesions. As I said, if some of these molecules are also, happening in the germline, it can actually pass through generations. From the parents, it can transform into the offspring, right? And this is happening because at one hand, the DNA is being damaged, and on the other hand, the same reactive species or this reactive nitrogen species, they can also compromise the ability for our germline repair. So repair is bad, mutations are more, and as a result of that, it becomes a cascading effect of transferring more mutations to subsequent generations. And we have been finding very exciting, at least pilot set of data that I wanted to share with you. So this is what I was trying to tell you, that if it happens across generations, it can create mutation load, and it can lead eventually to species extinction. It has a very strong consequence on adaptation and extinction process of species, right? So first thing that I can get into the methods later on, since I don't have much time, so I'll just quickly show you what we found so far. So this again, I experiment with tribolium beetles, and here the females have been assigned the ability or assigned the activity to fix the mutations which has been transferred into her from the male sperm, which has been mutagenized before. There are two groups of females. They receive this mutagenized sperm from the males, and they have the responsibility to fix those mutations. If they cannot fix the mutations, it will pass on to subsequent generations. And we found a very strong support of this entire thing here. So if you see that this is a beetle which have been infected with Bacillus thuringiensis, and if you look at the control populations versus the live infection population, you see a heavy increase in mutation transmission. And this activity we have been finding in other organisms also, like Drosophila, right? Here you see that, with respect to the sham infected population, the low infection molecules, low infection doses or high infection doses also have, higher mutation transmission. So it becomes a concern, as a novel cost of immune responses, which have not been figured out before this. And it looks like these observed effects are consistent across species. What we found, interestingly, this is the data where we have found, so the same evolution line that I have been talking to you about this, while they have been evolving to reduce the survival costs of infection, it seems the beetles have also evolved a better germline repairability. And they have now, transmitting lesser amount of mutation. This is exactly the data where I'm talking about. This is the typical thing that we would expect. If it is a sham infection, the control lines, if you infect them, very high rise of mutation transmission in the next generation, but when they evolve with, you see, even after being infected, the mutation transmission has not gone by. So, which suggests that besides, evolving immune system, organisms are also evolving their germline repairability. So, it is reducing the cost in multiple directions, so, and which gives us a sense of, how different things could be intertwined. I think I'm just going to stop here, and we are currently. Okay, yeah, so, and I think, we are just also, as I said, we have been a very preliminary stage of this experiment. We have found some interesting phenotypic data, and we are trying to fix or, find more information about how mechanism works. Besides understanding how immune response is evolving, we are also trying to understand how germline maintenance or DNA repairability has been evolving in these beetles. So, to summarize, so two things that I try to touch upon briefly, infection history, as a matter of whether it's a co-infection or a single infection, one could see divergent form of evolution against, divergent immune response against these pathogens. And the second thing that we found also, the evolution of germline maintenance to reduce the cost of mutation transmission. So I would just want to quickly acknowledge my lab members who have been putting a lot of blood and sweat to get this large-scale population-level data, and my funding bodies, India Alliance, Ashoka University, and SSC, the Study of Evolution Society in America, and IIT Delhi, to giving us some money to get some quick data on, do some RNA sequencing studies. Well, and we are accepting students for PhD, masters, and postdoc students. So if you're interested, please don't hesitate to reach me out. Thank you.
One or two questions from the students. Again, there'll be, the people will be available during lunch. First from the students. Let's give the students. Yeah. If the students don't have any questions, yeah.
All right, Imroze, very interesting work. I have one question. Was there a specific reason to select BT and Chirimanas Entomophila combination? Reason being that BT releases toxin, and Pseudomonas being an intracellular pathogen, there would be obvious differences in the way the immune system responding to that. And since BT is a more toxin-driven mechanism, why an immune system will play at a role, will really play a role in co-infection. So the reason is what is the basis of selecting these two pathogens for co-infection first? And second, given that they both act in very different ways, why do you think a toxin-related mechanism should give a very good contrasting feature to something like Pseudomonas, which is intracellular?
Actually, this is a very good question. I'm glad that you asked. So now both Entomophila and Bacillus thuringiensis, these are typically transmitted orally inside the beetle. So what we are doing here in this experiment, we are introducing this bacteria directly in the blood. So we are avoiding those toxin-producing pathways. So it is here we are enforcing two organisms to come together and fight. Right, so and it looks like the Entomophila and BT, they also had a lot of chemical ecology going in. So Entomophila, they releases a lot of hydrocyanic acid, and which kind of interacts with BT and try to kill. So we have been finding that if you have BT and P together, it looks like BT population slows down, it gets eradicated very fast. So there are a lot of interesting interactions going on, but we are not currently focused on toxic production. So we have been finding some interesting features about the different immune response being activated. So it is currently we're looking at the interaction between toll pathway and IMD pathway, which is again activated by these two pathogens. But yeah, what you said is also definitely one of the way to look at it as well.
But then you would say that since you are avoiding the gut, and the gut may be the first frontier that Phagocytosis uses to prevent infection.
Yeah, so sure. So, of course, because one of the reason being is a basically logistical reason, because for the running an evolution experiment, we have to ensure that for each individual, we try to minimize the error between how much individual beetle receives infection. So now when you induce, so we tried oral infection and we found that many beetles, they choose not to eat. So this behavioral avoidance towards infection makes us job very, very difficult. So we therefore chose to infect them directly, and this is a second most prominent way to get infected, because in a while they get injured, they keep on losing their arms, their the proboscis and all. So there are always hemolymph oozing out, and that creates a entry point for pathogen directly into the blood.
And endosymbions are there in Phagocytosis?
Yes.
Bacterial that may just like they have adapted to the
Of course.
insect. So do they contribute in any way to?
We have just tried to started scratching the surface. We are trying to look at the other microbiota present also. It looks like some of this immune response show dramatic impact by the microbiota. Some of this evolutionary response that we showed here, which I didn't show you in the slide, but removing microbiota somehow takes away this evolved reactions. It looks like even the microbiota, not only germline maintenance, not only immune response, it looks like if a microbiota has their own way to impact the immune responses, it becomes a very complicated altogether. So we just started our journey.
Okay.
Yeah.
So I just wanted to know, if you take the P-resistant population, excuse me, in the 21st generation, infect with BT, what would happen to that population?
They haven't evolved any cross resistance. It looks like...
No. I mean, would it be the same as like a virgin population, or would it be intermediate?
So you think the population which has evolved resisting is PE, they were infected with BT?
Mixed.
No, not mixed. I'm just saying, what would be the response first? Because probably the experiment is not done. If it is done, you can. If you take the P-resistant population, which should be isogenic after 21 sort of control meetings. So, is it resistant only to PE, or is there cross resistance or some kind of shadow?
Against BT, it doesn't show any cross resistance. So when we infected them with Bacillus thuringiensis, they don't show any response.
You have done that?
I have done that. Even with the mixed infection, they showed partial response to the thing. So the response is not as dramatic as the P would have been alone, but it somewhere falls in between. But there is a response against mixed infection also.
So evolutionarily, I think what she asked is relevant, that if the mode of infection, particularly which is gut mediated, could play a role in the evolution.
Of course.
So these two would not reflect the original question of evolution of immune system.
No, but here we are enforcing immune system to react, because blood is the site where all the immune reactions happen. So we could say that the immune response chances are more higher. In gut, many of the time infection cannot establish because we have gut microbiota. So any invading pathogen, they may not be able to colonize the gut, and they can easily create an infection.
But some of the immunity begins from the gut, otherwise it won't
True,
happen.
true.
Yeah, it won't happen otherwise.
Right. So, thank you, Imroze.
Okay, thank you.
Thank you.